And so what we did, being chemists, remember, we work with toxicologists, is we generated a series of C60s, which we basically were tuning in a very rational fashion. Fortunately, we're changing two variables about it, but the one I want you to focus on is this is C60. This is not C60. It would have a different molecular formula, but it's a close cousin. And as you go down this series, you're making a system that's a lot more soluble in water. Okay, and that's the primary tune. You're also making something that's less reactive. So there were two things we were testing. I'm not going to untease them here. But nevertheless, we asked the question, as the oil-water partition coefficient changes, in fact, by several orders of magnitude, do we see a subsequent change in toxicity? And in fact, you do. This is now, we're skipping systems. This is now going to mammalian cell culture. But what you see is actually the pristine stuff actually kills cells at very low concentrations. In fact, if you mapped this onto known organic molecules, it's actually a very acutely cytotoxic substance. However, if I hydroxylate it up, I make something that's virtually non-toxic. Now, this was really great data for myself and my colleagues to have in 2004, and you might not know why, but I'll share with you why. The reason was that asking questions about the toxicity of nanomaterials as a nanotechnologist was a very big challenge. You're coming from a community that wants its stuff to go out, to be used, to be applied, and here you are saying, well, you know, hey, this stuff might not be, maybe you should think about that. So we were actually getting a variety of interesting responses to both publishing the data and talking about the data in the nanotechnology community. So basically I was giving, and I like to use the word reframing because I learned it from your community. So I was out there sort of, you know, not positing this question at the beginning of my talks, but that's what people were hearing as I talked about doing toxicology studies. And that question was not one that was accepted or heard by the nanotech community. Might be a good way to say it. What that data that I just showed you allowed us to do was ask a very different question, but the same data set, which is how can we engineer safe nanoparticles? What things can we control that will give us materials that have less particular acute cytotox in this case? And that reframing was valuable. One of the things they did from a sort of widget perspective is one of the applications of C60 is as a dopant in polymer fuel cells, which we hope will replace all of the really nasty toxic batteries that are driving our laptops today. Those polymer fuel cells use C60 to help protons move through plastic. And you really need pristine C60, underivatized, to make that work. However, we knew that we could oxidize it, because that's basically what we do when we hydroxylate the surface, through the addition of a fairly mild to slightly less mild oxidant. So we came up with a packaging scheme that actually with our Japanese company collaborators is kind of an interesting idea. You would use the material in a polymer form in this pristine state. Before you dispose of it, you crack it. You release something that's a little bit tougher than bleach, and you take it to the hydroxylated stage. Now, that's not to say this hydroxylated material is one that I know a lot about, but at least on the obvious acute cytotox and the simple aquatic toxicity tests, we know it's a lot less reactive. So that's how you can use this kind of materials design to build in how you do the application. One of the things that the sort of dialogue of safety by design has done, which may or may not be interesting to this community, is it's created a little bit of a dichotomy in the research community. There's been a lot of discussion of the research strategy for nano EH&S. What kinds of data should we collect? And I like this because there's kind of two different paths. One path, which is the one I just talked about, bears a lot of resemblance to a drug delivery process, building sample libraries in which you control very precisely things about the material, running through some very exhaustive toxicity tests, and looking and actually using computation in a way that goes way beyond it's even used now in toxicology. So it's very computationally, quantitatively driven studies. Over here on this side is kind of the animal studies. You know, can you really get any useful data out of in vitro? Do you always have to do an animal study? And you can see right away the problem. If I'm working with 1 million libraries in high throughput screening, that's going to give me one kind of data set versus I'm going to, you know, place my bets that it's single wall nanotubes that are this long with this surface and this material, and I'm going to go do animal studies for 3 years. And that's really one of the dialogues that's developed in the policy community about which kind of data sets to collect. So that's been an interesting development. Clearly, both of them are very useful, but they do require different kinds of thinking about the research. Because predominantly because people's demand for, we want it to be relevant So e.g., we want to see a product already on the market. Problem is, I can almost guarantee the growth in the nanotech community is not going to come from products that are already on the market. So, you know, this one is good if you know the form of the silver and the exact state. This one is good for the future nanotechnology, which we know is going to dwarf the existing ones. Okay, so that's kind of one of the dichotomies that's been developed. I want to finally end with sort of some comments about communication, which is really, you know, bringing coals to Newcastle. But let me go ahead and tell you my thinking about it and what I've learned, at least personally, as a person who collects data and then has to talk about it with people. Well, this is sort of my first slide, which I've seen a lot. I love this slide because it really conveys something that as a widget maker, which is what I've been most of my career, I was used to creating a widget and proving to people that it solved a problem. But what I realized here, as I said earlier, is that my data is really only valuable because it helps this guy make a decision about how far he wants to be away from the tanker that's carrying nanoparticles. So really, my end user, my customer, is this person sitting right here. So the communications problem is critical for my research to have any impact at all. And if I don't do that right or don't figure out how to work with people who do it right, there will be no reason to take data on the kinds of stuff that I'm interested in. So it becomes extremely important. And in fact, I developed this chart because it's sort of analogous with tech transfer. So if you create information in a lab about, you know, how bad C60 is, or, you know, what kind of silver you want to use in your socks, that's new data. And if your goal is to have somebody make a useful decision, there's a lot of different things that have to go into it. And I've actually thought a fair bit about this from my sort of, you know, my perspective, not as informed as all of you. And what I concluded was that it's a much more complicated process than anticipated. And in fact, it was critical that I not make decisions as somebody who collected the data, which is kind of self-evident, but also that this sort of process of educating and framing the information is very laden with values, incredibly laden with values. So that as you go down this, and I do think it's a continuum, you begin to engage in more and more decisions and processes that begin to sort of pit you in a certain place with respect to people's perceptions of how you're communicating and what you're communicating. The other thing I learned is that there is no organization that does this. There's no mechanism, process, or way for me to do that. So one of the things that is most challenging in this picture is actually the fact that the technical data is, I'm not going to call it a mess because that's not fair to a lot of my colleagues, but it's close to that. It's really, really hard to take our materials and study their biological properties. It's just a hard scientific problem. And hard scientific problems do not get solved by one person doing one experiment, even like a whole bunch of smart people in 5 years. This community will start to have its act together probably in about 3 to 5 years. We're still trying to like make sure my lab gets the same data as the lab in Ireland does with the same materials and the same cell lines. It's a very hard set of stuff to do. And because of that, if you go to the papers about C60, which I've done, I've started to do my own meta-analysis. So the early papers out of CBEN on C60 impacts have actually well over 100 citations now, which by the way is good for that's a lot of citations, widely cited. There's about 4 key papers. If you go and you look at those papers, half of them more or less agree with the technical findings, but add to them, modify them. The other half are violently in disagreement. And that is related to issues of characterization, how you do the experiment, lots of subtle things I'm not going to go into, except to tell you it's normal. It's normal in science that this would happen. And sorting it out is what we do. We're sorting it out now. So no single paper or study can be definitive. That's not to say we're not lowering the uncertainty and beginning to find our way. It's just very early stage. So you have to go through this communications process when the data is not conclusive. And that's different, I think, than uncertainty in some important fashion. Because when you're uncertain, you can be conclusive about your uncertainty. When the data's all over the map, it's just kind of all over the map. Okay, so one of the things that we were compelled to do early on was we realized when you have a lot of conflicting technical data, it is really easy for people to hijack the technical data and use it to make values arguments, but sound like they're making technical arguments, which really bothered a lot of us. So one of the drivers for creating the International Council on Nanotech was actually to try to get multiple stakeholders to communicate information so that the odds of me looking at the technical data and coming up with my particular view of it